So this is part 2 of the Chapter 13 PowerPoint lecture. And we left off just talking a little bit about these fluorescent karyotypes. And I wanted to point out how you can see very clearly with the fluorescent dyes the homologous chromosomes. And I wanted to remind you, when you're looking at homologous chromosomes, what exactly are you looking at? So we have these pairs of chromosomes that are carrying the same genetic information in terms of the same genes. So they're coding for the same DNA traits and the same genes, but they're not identical because one of them has come from the maternal parent and the other has come from the paternal parent. So one from the mother and one from the father. And so that's how we get our variety of genes in the offspring, because we have a different set of genes coding for the same traits in from the mother and one from the father. So homologous chromosomes, again, are paired chromosomes. They both carry a pair of matching genes, and they control the same inherited characteristics or traits. Homologous comes from homo, meaning the same, and logos, meaning information. And wow, there was supposed to be more on that slide. That's interesting. Oh, there it is. So diploid, as a reminder, we use the insignia 2N to talk about diploid, which means we have two sets of chromosomes. So one from one parent and one from the other. That's how we end up with two. So here we have the black, let's say, being from the mother. So we have one of this homologous pair, and here's the other one of the homologous pair. So this is from the mother, this is from the father. And then here we have the short one from the mother and then the matching one from the father. So single-stranded homologous chromosomes. And these little bands show you, for example, a locus of a gene. So we would have the same genes at the same locus on the different homologous strands. And then, not to confuse you, but when we go through replication of DNA, before we start the process of meiosis or even the process of mitosis, what we have is a replication of each of these strands of DNA. So we end up with two strands of the father's and two strands of the mother's DNA. And this is still a set of homologous chromosomes, but now they're double-stranded. And actually, I don't even like that word, because when we're talking about a double helix of DNA, I don't want you to confuse that, because this is a double helix of DNA, just this one strand here. But what is a better term here would be to say that these are the sister chromatids of the same chromosome, and these are sister chromatids of the same chromosome, and then these two are actually homologous chromosomes. And then here's one final picture talking a little bit more about a karyotype using fluorescent dyes. And this is even a better photograph, because if you zoom in here and look, you can see the really clear banding of the different genes located on each of these chromosomes. This is a very nicely dyed karyotype. And then this would be a close-up showing what you see here. So in this case, we have one homologous chromosome from mom, one from dad here. And so here we have the one from mom and one from dad. And if you look at it even more closely, this is occurring this picture is taken during metaphase. So we do have the sister chromatids already present, because before we enter mitosis, we've already gone through replication. Or before we enter meiosis, the chromosomes have gone through the process of making sister chromatids. So you can see here, this is actually two strands of the same chromosome joined together. So each of these homologous pairs is actually a set of sister chromatids. And you can see here the centromeres. Again, another thing to keep in mind with homologous chromosomes is that they're going to have the same staining pattern, they're going to have the same centromere position, and they're going to be the same length. Okay, so this is really repeating what we've been saying all along, but just for clarity, I don't think it's harmful to keep going over it. But a diploid cell, again, is represented by the 2N, and that means there are two sets of chromosomes. And so for humans, the diploid number is 46. So our 2N equals 46. But the haploid for a human, the N, is going to be exactly that, half of 46, or 23. And haploid means you have one copy of the genetic material, and when we have diploid, we have two copies of the same genetic material, or sets of genes. And the only place in humans we have haploid cells is in the gametes. And all of the rest of the cells, which are called somatic cells, so all of our body cells, are diploid. So this is a picture that's showing how we have our sister chromatids. And here, this is the paternal set is blue, the maternal's red. And in this case, the 2N equals 6. So the diploid total number of chromosomes for this cell is 6. So if you count this here, we'll just make sure we have the right count. We have 1, 2, 3 from the father, and 1, 2, 3 from the mother for our total of 6. So the maternal set has an N of 3, and the paternal set had an N of 3 also. And each of these chromosomes, this is one chromosome, but it's actually a set of two sister chromatids because we've already gone through the duplication process. But the homologous chromosomes, these two are the same size, so they're the same set of genes. So these are a homologous pair, and they're not identical. They are non-sister chromatids, these two chromatids here. So the human life cycle begins when a haploid sperm cell fuses with a haploid ovum. And what we're going to get then is, let's see if we get this to work, is a fusion of the nuclei. And that fusion process is called fertilization. But in order to get these haploid sperms and eggs or ova is the process of meiosis. So it's only through meiosis that we can go from a cell that has 46 to a gamete that has 23, or half that number. So to go from a 2N to an N is only going to occur through meiosis. And then when you have fertilization, these two will add up together because we have a fusing here of these two cells in order to go back to the diploid or 2N state of 46 with our zygote or developing embryo. So meiosis is the process by which we're going to produce gametes. And we have, similar to what we discovered with angiosperm reproduction, we have alternating stages in the production of gametes. So the chromosome number has to be reduced through meiosis from diploid to haploid, or 2N to N, or 46 to 23 in humans. So that process occurs through meiosis. So we start here. This is our diploid stage. That's one stage. And our second stage is a haploid stage. So we have the development here. This is a zygote or an embryo that is through mitosis, grows and develops into a full-grown adult. And then the adults, in order to produce haploid gametes, and those are produced in the male in the testes and the ovary of the female. In order for those to be producing what we call sperm and eggs, that process of producing gametes is through meiosis. And so meiosis allows that diploid set of cells to actually produce haploid cells that, when fused in fertilization, will again produce a new diploid zygote or offspring. So for this reason, we call meiosis reduction division, because it's going to reduce the chromosome number from 2N to 1N, or diploid to haploid. So one thing, I think it's pretty obvious, and I'm sure as a review, because this is not the first time that you're being or learning and reading about this topic, but the stages and machinery of meiosis and mitosis are very similar. So it can often be confusing if you don't stop and think about what are the differences. So we're going to make sure that you don't confuse the two in any way. So again, talking back a number of slides ago, we were talking about sexual life cycles. And there are three main types of sexual life cycles among living organisms. So, and what differentiates the three is the timing of meiosis and fertilization. So how long meiosis is part of that life cycle and how long fertilization is part of that life cycle. Or how long is the organism spending its lifespan is a 2N, a diploid organism, or a haploid. So starting with animals, which is what we're really focusing mostly on here, we already talked about how the adult here is 2N, so it's a good portion, the largest portion of the three life cycles that we have here, that is spent 2N. And then through meiosis, we have the production of gametes, and then when two different gametes are joined together in fertilization, we end up back from haploid back to diploid. And we just studied the plant angiosperm in particular life cycle, and we saw how that also has two different stages. So we have the alternating sporophyte and the gametophyte stages. And so during the sporophyte stage is when the plant spends, again, most of its lifespan as an adult, and during that time period it's diploid. So the sporophyte plant stage is the diploid stage. And then through meiosis, then the sporophyte plant produces spores, which are then haploid. But the plants, as you guys will remember, is a little more complicated. It's not as simple as just producing a haploid gamete and then joining with another, such as we see in animals. But in a plant, these spores actually have to go through more development before they actually become gametes. So the gametes are produced through mitosis. So the spores going through mitosis then become gametes. And then we have, again, the fusion of two different gametes through pollination if it's an angiosperm, and we get the fertilization and the return back to a 2N or sporophyte stage.